What is RNA polymerase, and what is its primary function in cellular processes? RNA polymerase is an enzyme responsible for synthesizing RNA from a DNA template during transcription. Its primary function is to transcribe genetic information from DNA to RNA. Hello, I'm Luna, the Wise Owl Scientist. Today, we're diving into new concepts Luna style, making them not only interesting but easily understandable. So, without further ado, let's get started and explore this exciting journey together. How many types of RNA polymerase are found in eukaryotic cells, and what are their specific roles? Eukaryotic cells have three types of RNA polymerase: RNA Pol-I, RNA Pol-II, and RNA Pol-III. RNA Pol-I synthesizes rRNA, RNA Pol-II transcribes mRNA, and RNA Pol-III transcribes tRNA and other small RNAs. Describe the basic structure of RNA polymerase. RNA polymerase has a multi-subunit structure. In prokaryotes, the core enzyme consists of 5 subunits, while eukaryotic RNA polymerases have more complex structures with additional subunits. What is the function of the sigma factor in bacterial RNA polymerase? The sigma factor is required for the initiation of transcription in bacteria. It helps RNA polymerase recognize the promoter region on DNA and facilitates the binding of RNA polymerase to the promoter. How does RNA polymerase know where to start and stop transcription? RNA polymerase identifies the start site by recognizing the promoter region on DNA. Termination signals, such as specific DNA sequences or structural elements, signal the end of transcription. What is the role of the TATA box in eukaryotic transcription? The TATA box is a DNA sequence found in the promoter region of many eukaryotic genes. It helps position RNA polymerase II on the DNA strand and is crucial for the initiation of transcription. Explain the difference between the coding strand and the template strand during transcription. The coding strand has the same sequence as the RNA transcript, while the template strand is used by RNA polymerase to synthesize the complementary RNA molecule. What are the similarities and differences between prokaryotic and eukaryotic RNA polymerases? Both types of RNA polymerases share the general function of transcribing DNA to RNA. However, eukaryotic RNA polymerases are more complex and involved in the transcription of different types of RNA. How does RNA polymerase proofread and correct errors during transcription? RNA polymerase has a proofreading activity that allows it to backtrack and correct errors in the synthesized RNA. This enhances the accuracy of transcription. What is the role of transcription factors in eukaryotic transcription? Transcription factors are proteins that regulate gene expression by binding to specific DNA sequences and interacting with RNA polymerase. They help initiate or enhance the transcription process. Can RNA polymerase perform de novo synthesis, and what does this term mean? Yes, RNA polymerase can perform de novo synthesis, meaning it can start synthesizing RNA without requiring a pre-existing primer. How does the process of transcription differ between prokaryotes and eukaryotes? In prokaryotes, transcription occurs in the cytoplasm and is coupled with translation. In eukaryotes, transcription occurs in the nucleus, and the mRNA is then transported to the cytoplasm for translation. What is the significance of RNA polymerase II in eukaryotic cells? RNA polymerase II is responsible for transcribing mRNA, which serves as a template for protein synthesis. It plays a central role in gene expression in eukaryotic cells. How is the activity of RNA polymerase regulated? RNA polymerase activity is regulated by various mechanisms, including the binding of transcription factors, chromatin structure, and post-translational modifications of the polymerase itself. What are the consequences of errors in transcription, and how are they addressed by the cellular machinery? Errors in transcription can lead to the production of faulty proteins. The cellular machinery employs mechanisms like RNA degradation and quality control to eliminate defective transcripts and maintain the integrity of gene expression. Unraveling the molecular symphony of RNA polymerase, we explored its diverse roles, structural intricacies, and regulatory ballet, delving into the dance between transcription factors, initiation precision, and the elegance of proofreading, ultimately revealing a choreography crucial for the cellular narrative. In this molecular odyssey, from de novo synthesis finesse to the consequences of errors, each question unveiled a dynamic interplay, showcasing RNA polymerase as the maestro orchestrating the genetic score in the cellular opera. Thank you so much for watching. I appreciate your time and support. If you enjoyed this video, please consider subscribing to our channel and ringing the notification bell so you won't miss any of our future content. Your support means the world to us, and we can't wait to share more exciting videos with you.